Hi everyone, my name is Luz Serrato-Diaz. Today I'm going to talk about coffee rot in Puerto Rico, new associations with Colletotrichum, Fusarium, and the coffee berry borer. What is known about coffee fruit rot? The disease can be divided into coffee bear disease and anthracnose. And according to the literature, coffee bear disease is caused by Colletotrichum kahawai that affects green coffee berries in Africa, while anthracnose affects red coffee, and different species of Colletotrichum have been reported worldwide, including in Puerto Rico in 1985. How do we know about coffee fruit rot in Puerto Rico? Well, this occurred when I was studying my doctorate working with Bouveria bassiana, a biological control of the coffee berry borer. One day in the field, I noticed that the perforated fruits by the coffee berry borer had a necrosis. So some farmers and extension agents said that it was sun damage. And as a pathologist, I say no, I don't believe it. This is a disease. I noticed that green and red coffee were affected, and at that moment I did a literature review and I saw the two diseases that I mentioned it in the previous slide, coffee berry disease and anthracnose. So that made me think if there is an interaction between coffee berry borer, Bouveria bassiana, and coffee fruit rot. To answer that question, we collected 2,333 coffee fruits perforated by the coffee berry borer. Half of them with Bouveria, the other half without Bouveria, in 3 locations in Puerto Rico, Adjunta, Ciales, and Utuado, and we evaluated fungal diversity and percent of fruit rot. We found from this survey that fruits with Bouveria had significantly less coffee fruit rot than fruits without Bouveria. And when we plated all coffee fruits, rotten coffee fruits on petri dishes on PDA, we found different fungi, including Bouveria bassiana, different species of Colletotrichum. As you see in this picture, there are different colors. These are different species of Fusarium and different species of Colletotrichum. With these results, finding Colletotrichum and Fusarium in the petri dishes made me think the new scientific questions. The first one, which pathogens cause coffee fruit rot in Puerto Rico? Are all of them Colletotrichum? How many Colletotrichum species are interacting? Because we found that fruits with Bouveria had less coffee fruit rot than fruit without Bouveria. Can Bouveria bassiana be a potential biological control against coffee fruit rot? Is the coffee berry borer playing a role as a dispersal agent of coffee fruit rot? Because several Colletotrichum species have been reported worldwide causing either coffee berry disease or anthracnose, the first thing that we did was identify Colletotrichum species. We found 4 Colletotrichum: Colletotrichum siamense, tropicale, teuromicola, and fructicola. One thing is found Colletotrichum in tissue, but the other thing is to say that Colletotrichum is a pathogen. So we did pathogenicity tests with all 4 species of Colletotrichum found. As you see in the figure on the left, in y-axis is the percent of external rot, and in x-axis is the percent of internal rot. The crosses in the graph represent the percent of internal and external rot for each isolate. For example, the red cross represents the percent of external and internal rot of Colletotrichum tropicale. This isolate causes more external than internal rot. You also can see this in coffee fruit shown on the right. Colletotrichum tropicale causes more external than internal rot. Colletotrichum teuromicola causes more internal than external rot. Colletotrichum siamense causes both, and so on. We discovered with this test that all species can cause coffee fruit rot, and there was an ecological niche for each species of Colletotrichum. Because we found that coffee fruits with Bauberia had significantly less coffee fruit rot than coffee fruit without Bauberia, as is shown in this graph, can be Bauberia bassiana a potential biological control against coffee fruit rot? To answer that question, we conducted experiments in the lab and in the field. In lab experiments, we had 5 treatments. Treatment 1, perforated fruits plus Bauberia bassiana. Treatment 2, perforated fruits. Treatment 3, no perforated fruits. Treatment 4, perforated fruits plus Colletotrichum, our positive control. Treatment 5, perforated fruits plus Colletotrichum plus Bauberia bassiana. As is shown in figure A on the left, x-axis is showing the treatments and y-axis is showing the percent of fruit rot. Treatment 5, fruits that we perforated, and 2 days before Colletotrichum inoculation we put Bauberia bassiana. The percent of fruit rot was significantly less than the positive control treatment for perforated fruit with Colletotrichum alone. When we played it on PDA, PDA is a media to grow fungi. In treatment 4, we found Colletotrichum. You see in the petri dishes, the gray color is Colletotrichum. And in treatment 5, we didn't find Colletotrichum. So we say, wow, we did it. We control coffee fruit rot in Puerto Rico. Now, let's test this in the field. In field experiment, we had the same treatments that we had in lab experiments. Again, the figure C on the left shows in x-axis the treatments and in y-axis the percent of fruit rot. Treatment 5, perforated fruit that we put Bauberia 2 days before Colletotrichum inoculation, had significantly less percent of fruit rot than our positive control treatment 4, perforated fruit plus Colletotrichum. However, treatment 1 and treatment 2 had the same fruit rot than our positive control treatment 4. Remember, the same letters in the graph means that there is not a significant difference. At this moment we say, oh my god, this didn't work in the field. But when we put all these treatments in petri dishes to see what was happening, in treatment 1 and treatment 2 were found Fusarium, the red color that you see on the right in petri dishes. With the discovery of Fusarium in our petri dishes, we formulated another scientific question. Can Fusarium also cause coffee fruit rot in Puerto Rico? We published the role of Fusarium in 2024, and we found 9 different species of Fusarium in 6 different Fusarium complexes. Fusarium solani species complex, Fusarium sambucinum species complex, Fusarium oxysporum species complex, Fusarium concolor, and Fusarium lateritium. Again, one thing is found Fusarium in tissue, but the other thing is to say that Fusarium also was causing coffee fruit rot. So we did pathogenicity tests with all the 9 species found, and only 5 species cause coffee fruit rot in green coffee in Puerto Rico: Fusarium bostricoides, Fusarium solani melangonae, Fusarium nirenbergiae, Fusarium pseudocecinatum, and Fusarium lateritium. In general, the 5 Fusarium species cause more internal than external fruit rot. Because we found in the field coffee fruits perforated by the coffee berry borer with necrosis, we thought that maybe the coffee berry borer is playing a role as a dispersal agent of coffee fruit rot. To answer that question, we conducted lab and field experiments. In lab experiments, we had 4 treatments. Treatment 1, the coffee berry borer with Colletotrichum. Treatment 2, the coffee berry borer without Colletotrichum. Treatment 3, non-perforated fruits. Treatment 4, perforated fruits with Colletotrichum. This is our positive control. To do these experiments, we impregnated the CBB, the coffee berry butter, with a solution of Colletotrichum spores, and we let them dry for 2 hours, and then we place 40 females of the coffee berry butter on coffee branches with at least 30 coffee fruits. As is shown in figure on the left, in x-axis are the treatments, and in y-axis is the percent of fruit rot. Treatment 4, our positive control, perforated fruits plus Colletotrichum. This already we know that they cause the disease. And for this reason, you see the big bar and almost 100% of fruit rot. But if you see treatment 1, the coffee berry butter impregnated with Colletotrichum also cause the disease. That means that the coffee berry butter can disperse Colletotrichum in the laboratory. With this in mind, we conducted experiment in the field, but this time we impregnated the coffee berry butter with the 4 Colletotrichum species. As is shown in both graph on the right, all the Colletotrichum species, this is the different colors of the bars of different colors of in the figure, cause internal and external fruit rot in the field. Again, one thing is to say that the coffee berry butter can disperse Colletotrichum, but the other thing is to say that the coffee berry butter has it. So to know which fungi are part of the coffee berry butter microbiota, we plated on PDA 630 coffee berry butters collected from the field. Half of them were disinfected with a solution of 70% of ethanol, and the other half were not disinfected. Disinfected coffee berry butters were smashed, and as much of the skeleton, including antenna and legs, as possible was removed and plated on PDA. From this experiment, we recovered 158 isolates, where the most common genera was Fusarium. 5 Fusarium species, Fusarium bostricoides, Concolor, Lateritium, Meridionale, and Pseudocircinatum, were found as part of the coffee berry butter microbiota. But only 3, Bostricoides, Lateritium, and Pseudocircinatus, cause coffee fruit rot in Puerto Rico. The graph on the right showed the experiments with Fusarium and the coffee berry butter. The black bars without stripes represent the percent of fruit rot, internal and external, of the treatment that when the coffee berry butter was impregnated with Fusarium. In both cases, internal and external fruit rot, the coffee berry butter impregnated with Fusarium were able to cause the disease. Because farmers and extension agents were saying that the necrosis on coffee fruit of certain fields was caused by some damage, we conducted a survey to know if there is an association between sun coffee and coffee fruit rot. The graph on the left shows the incidence of coffee fruit rot in sun, intercalated, and shade coffee. We found that sun coffee had significantly more coffee fruit rot than intercalated and shade coffee. The graph on the right shows the negative correlation between altitude and coffee fruit rot. That means higher altitudes in Puerto Rico, we got less coffee fruit rot. From this survey, we also found that internal rot was caused mainly by Fusarium species, and external rot was caused by Colletotrichum species. In summary, Colletotrichum species were found only in rotten coffee tissue, but not as part of the coffee berry butter microbiota. The 4 species of Colletotrichum found were pathogens to coffee, causing coffee fruit rot. In general, 9 species of Fusarium were found from surveys. However, only 4 species were found in coffee tissue, and 2 of them, Solani melanogae and Nerium berghei, were pathogens to coffee. 3 Fusarium species were found in both rotten coffee tissue and as part of the coffee berry border microbiota. All 3 species were pathogens to coffee: Fusarium bostricoides, Pseudocircinatum, and Lateritium, where Fusarium bostricoides was the most common species found in both rotten coffee tissue and from the coffee berry border. In general, sun coffee had more coffee fruit rot, and in more altitude the coffee fruit rot was less. With all this information, now we can answer the scientific question that we formulated in the beginning. The first one: which pathogens cause coffee fruit rot in Puerto Rico? Colletotrichum and Fusarium. Are all of them Colletotrichum? No, there is also Fusarium. How many species of Colletotrichum are interacting? 4 species of Colletotrichum. How many Fusarium cause coffee fruit rot? 5 species of Fusarium. Is the coffee berry border playing a role as a dispersal agent of coffee fruit rot? Yes, the coffee berry border is the dispersal agent of Colletotrichum, and it has Fusarium as part of its microbiota. And can Bouveria bassiana be a potential biological control against coffee fruit rot? Yes, when the infection is caused by Colletotrichum. Bouveria doesn't work when the disease is caused by Fusarium. Bouveria has a dual purpose: controlling the coffee berry border and indirectly reducing coffee fruit rot when the CBB is controlled. The current work that we are conducting at TARS, we are testing or screening the 31 IMLBT International Multi-Location Variety Trial coffee varieties from the World Coffee Research against coffee fruit rot. So far, we have evaluated 18 coffee varieties, and we found one of them with potential tolerance to coffee fruit rot. Thank you for your attention. If you have any question, I'll be taking them at this moment.